Welcome to our first fully vodcasted lecture for this semester. If you're joining me from the classroom, it is Friday, February the 1st, and you're probably beginning to think a little bit about your exam coming up. So we do have that exam on Tuesday the 5th, and as we talked about last time, a review has been posted on Doc Sharing, and you can find that there. And also I'll hold those review sessions on Monday, February the 4th. The one will be from 2 to 3 p.m. face-to-face, and that's the one that will be held in the room right next door to the lab. And then I'll do the Illuminate session online. And as I mentioned, I'll try to also record that session, technology willing. So we talked about the format of the exam last time and the fact that there's 25 multiple choice that you will answer as half of your grade, potentially, because I'll actually weight more heavily either the multiple choice or the free response. So that gives you the best result depending upon which section you perform the best on. But then on question number 26, everyone will be asked to answer question 26 in regards to gram-positive and gram-negative cell wall structure. I promise that you will have to draw both, so you may want to think about bringing with you your awesomely colored pencils or pens or something that you can use to make your drawing really give it a lot of character and a lot of color, whatever you want to do to show me that you know the structure of gram-positive and gram-negative cell walls. So for that question, number 26, everybody has to answer that one, which means that you then need 4 more questions to pull together what is a potential of 50 points from the free response sections. So for questions 27 through 32, you will choose to answer only 4 of those questions. Might be that one of those choices is Omnivore's Dilemma. You can choose that if you participated in the discussion. If not, you'll want to choose 4 other questions and tell me which one you chose by circling your questions. Or you can also X off the questions that you didn't choose to do. Whatever is very clear to me to show me which ones you did do, and that way I can really tell which ones you want me to grade, and I don't get confused grading one that you don't want me to grade. So I think that summarizes the most important announcement for the day. Do finalize your poster topics if you have not already done so, and hey, let me know if you need any help at all as you continue your journey on your posters, trying to decide on how to narrow that topic a little bit. A lot of that will just be to do more reading and find out more information about your topic. Gram stain report due on Thursday, February the 7th, so that'll be our first lab report. It is very short, very succinct, should be a very quick write, but make sure you follow the assignment description and the advice of your TAs as given when they assigned the report. So I'm going to dive in because I am totally stoked about talking about structures external to the cell wall, because when we left off last time we had talked about the gram-positive and gram-negative cell wall. So now we might want to look just a little further out. So outside of the most outer layer of the cell wall, some bacteria have an additional layer. That additional layer often plays a very protective role for the cell, and in many cases it may also play a role in adherence to certain surfaces. One of the types of outer external layers is called Glycocalyx. Glycocalyx is usually typified as being a polysaccharide layer. So you can see how it'd be like a thick, well-hydrated, literally coat on the outside of the cell. I sometimes think about it that way. It's like putting on a coat when you go outside. It's very protective. But of course this coat is saturated in water, giving these cells a slickery sort of characteristic. And on an agar plate, as Jennifer reminded us last time, that would make them look very mucoid on that plate. So in addition to having a lipopolysaccharide layer in gram-negative, sometimes they also add this layer of glycocalyx that makes them look even moister and more mucoid on the surface of a plate. So glycocalyx functions in either protection or attachment. And we can actually split down into 2 different types of glycocalyx. The first type is called a capsule. And a capsule is super well organized and very distinct. You can actually see it when you look for it. So in lab, during lab 4, we're actually going to look at a bacterium called Klebsiella Pneumoniae that forms a really distinct capsule. And we'll put it under the microscope. Of course, we won't heat fix it because as Tyler would tell us, that would shrink it all up, right? And we wouldn't be able to see the capsule. But we will stain it so that we can see this coat outside of the bacterial cell. And we'll actually be able to see it looking like a halo around the cell that it protects. So a capsule is one type of glycocalyx, and you can see in the drawing down here below I've summarized these. So capsule being one type of glycocalyx, glycocalyx being one type of layer external to the cell wall. Generally it's a polysaccharide layer. The other type of glycocalyx, you can see here, is a slime layer. So let's talk a bit about a slime layer. The thing that distinguishes it from a capsule is that it is very diffuse and irregular. And sometimes it's not possible to observe it in the lab. Sometimes we will see it maybe by almost default, and sometimes we'll see it in vivo. That is to say, inside the body. For example, Staphylococcus aureus, right, the causative agent of staph infections, quote unquote. That guy forms a slime layer in vivo, in the body. And so it's a little harder to observe. We can't really put it on a microscope very well and see it, but it is still a very protective layer. In lab, we're going to talk a little more about the specific functions of a capsule, but suffice it to say that it protects the bacterium. So if it keeps the bacterium from being attacked, for example, by a phagocytic white blood cell or by an amoeba cell trying to eat it and engulf it. It can also protect the cell from hydrophobic toxins that don't want to pass through that really water-filled layer. So it's a very protective sort of layer. Also, we'll talk about how bacteria sometimes that are on your teeth, which will fascinate a lot of you guys headed for dental school, will recognize that sometimes those bacteria, for example, Streptococcus mutans, actually adheres to your teeth by using a capsule layer that it has very well developed on the outside. So a lot of functions of glycocalyx. Let's look at the next type of layer external to the cell wall. This is the S layer. And an S layer is very different from a glycocalyx because it is made of, for one thing, it's made of glycoprotein or protein. So it's got a different macromolecular composition. So this is one thing that distinguishes it. But another thing that actually distinguishes it, and I'm actually going to go ahead and draw this, is that it has sort of this floor tile-like pattern. So if you think about it maybe as either squares or hexagons or what have you, the way that floor tiles are sort of patterned, I'm trying to draw that there, and you can picture another one down here, another one down here, making this very even repeating sort of structure. And that's why this is actually going to fascinate a few of you. For example, I was thinking about Paige and Trevor and Kyle and all of you chemical engineers, maybe looking at biotech applications. But also Keenan specifically told me that he was interested in biotechnology. And S layers are used a lot in biotechnology because of the evenly repeating floor tile-like pattern. They form these specifically sized pores, about 2 to 8 nanometers, and they can be used as membranes, like biomimetic kind of membranes. So I'm actually going to show you this particular article. It's called Biotechnology and Biomimetic with Crystalline Bacterial Cell Surface Layers, in parentheses S layers, specifically S layers. So this is an article in Micron, and it gives us an idea of the fact that these are really used a lot in biotechnology. Caulobacter crescentans is one that forms an S layer, and it is often used in bioreactors and biosensors. So I thought that would interest all of you with those particular types of interests. So a regularly and distinctively structured layer of protein or glycoprotein, common amongst both bacteria and archaea, so more than one domain of life. May protect against pH fluctuations, osmotic stress, enzymes, and predaceous bacteria. This is the coolest thing, right? When you have a bacterium that likes to literally eat other bacteria. There is one called Bella vibrio, and this is like totally violent, but what it does is it comes and it winds itself up using Flagella Rotation, and it smacks into a prey cell and bores a hole into it. Well, if a cell has an S layer, that floor tile-like pattern would protect it from the boring of this predaceous bacterium. So these also may help maintain cell shape, rigidity, promote adhesion, protect against phagocytosis. So this is also the coolest thing, because picture a phagocytic white blood cell coming up and trying to engulf a prey bacterium. if it has a capsule on it, it's literally going to slip out of the grip of the white blood cell or of the amoeba trying to catch it. So it's literally a slippery little sucker that's going to just get away from that engulfing process that we see in phagocytosis. So pretty cool to have either, if I were a bacterium, I would totally want to have an S layer because it's sweet looking. It's like, it'd be kind of like having like a sweet, snake skin jacket that's really highly patterned, and it protects you. It's very, very protective. The last outer layer that I want to talk about is called a sheath. We're going to talk more about sheaths later when we talk expressly about bacteria that live in aquatic environments. But what a sheath is, it's kind of like a straw-like structure that surrounds a community of bacterial cells. And you guys are going to laugh at me, but I totally wanted to show you a demo of what this would look like, to an extent a model, and so I busted into the lab and totally defaced one of the tubes for one of the bottles in the lab, and I thought this is a way that I could show you what a sheath is like. So if you picture these beads in here as being the bacterial community, there's a lot of bacteria packed in together inside of the sheath. And the sheath surrounds it and allows for a community of cells to be encased in this sort of straw-like structure. So this is protective because the sheath itself can be adhered to surfaces. The bacteria can then, if they're in an aquatic environment, you can see how water's flowing by, and as the water flows by, they can capture the nutrients that come with that flowing water. It protects them from predaceous bacteria like Vibrio. So all in all, it's a sweet deal to have a sheath surrounding a community of cells. And we'll even learn that bacteria can actually divide labor inside of a sheath structure. So some bacteria may differentiate to do one job versus others that may differentiate to do another job. So who said that prokaryotic life was simple, right? All right, let's take a look at something else that's also external to the cell wall. These are the flagella, and we know that this is only found in bacteria that are motile. But flagella are long hair-like protein structures responsible for motility. And when we look at this cheesy drawing that I've done of a flagellum, you can look and see how the flagellum is actually anchored into the cell wall of this bacterium. And I hope that as you're looking at this, I'm totally picturing Olivia saying, hey, that's a gram-negative cell wall. And I can tell because there's an outer membrane, there's an inner membrane, there's a periplasmic space, and a very thin layer of peptidoglycan. I also see the LPS on there. So this must be a motile gram-negative bacterium, and we can see how the flagellum is rooted into what we call the basal body of the flagellum is rooted into the cell wall. So these flagella, I can go ahead and label some of the structures on here, can be split into three separate segments. Right here, this is the filament. The filament is what's out there actually leading to the motility, because it actually is what will be sort of flapping in the wind, if you will, as the rotation takes place. And that's how bacterial flagella move, is to rotate. So they'll rotate either clockwise or they'll rotate counterclockwise. So that filament will move with that corresponding rotation. The filament is made up of a protein called flagellin. That makes a lot of sense. Flagellin makes up flagella. So the flagellin protein, it actually spins around and forms a hollow core inside of the filament. So it's a very, very suited to its function in that way. So the next section of the flagellum is this one here, called the hook. And you can see why. The hook is the structure then that allows for either rotation clockwise or rotation counterclockwise. Down at the very bottom and secured into the cell wall, this whole region is called the basal body. And we can further distinguish parts of the basal body. For example, you have the L ring. This is the P ring. Down here we have a couple of additional rings. This is the MS ring. And then finally the C ring. These used to be called the S&M rings, and I'm pretty sure they stopped naming it that because I was making dirty jokes about it stabilizing sort of whip-like structure, but anyway, that's neither here nor there. So let's look then at how this flagellum moves. So as I said, spinning either clockwise or counterclockwise in a very rotatory sort of way, but it uses the PMF. So remember that that stands for the proton motive force. So as protons swoosh into the cell, that downward influx of protons drives the flagellar rotation. And there's some complexity with that. There are some apparatus, I should say, that do line next to the basal body and allow for that influx of protons. And there's some complexity in how that works. But essentially we know that the PMF is tapped into as the energy source for flagellar rotation. So we can note that they move like propellers, and holy cow, they move fast. So I know that Grant is a runner, and you really like to, get out and go fast on occasion, I imagine. But here's the thing. In bacterial cells, they can often move the flagella at as fast as 1,000 revolutions per second. For example, a bacterium called Vibrio alginolyticus is able to move that fast. So now, if Grant were able to move as fast as a bacterium, he would be able to run 82 miles per hour. Now, Grant, I know you're fast, but not even Usain Bolt comes close to that fastness, right? It's not even a contest. Bacteria are way faster. But don't worry, we have some things up on them because unlike us, they cannot sense spatially. So even though they run really fast, so to speak, they move around very quickly, they don't ever move directly. That is, they do a lot of back and forth to get to somewhere. We'll talk about how that works in a minute. The other thing that we should say about flagella is that sometimes they are virulence factors. That is, sometimes they enhance the pathogenicity of a cell. And in fact, that's a conserved theme today, isn't it? Because we know that structures external to the cell wall, like, for example, the capsule, also increased virulence because they allowed the cells to escape from the phagocytic white blood cells, so increasing disease-causing potential. A good example of something that has increased virulence because of its flagella is Helicobacter pylori. And this is the one that I showed earlier in class. It has flagella in a tuft at the top of the cell. Some of my students at one point gave it a gangsta headband, which is only partially legitimate because, in fact, Helicobacter is both good and bad in our gut. We know that it can be the causative agent of ulcers, peptic ulcers, but also in the right amounts, it can help protect you against things like acid reflux. So this is a double-sided bacterium, so to speak, but it does utilize its flagella and flagellar rotation to bore into the stomach epithelial lining, thus increasing its virulence potential in causing peptic ulcers. So let's move along and talk about the types of flagella, that is to say, how they can be distributed around a cell. And sometimes bacteria only have one flagellum. We call that monotrichous. Monotrichous, single, right? One flagellum. Sometimes that single flagellum is located on the end of a cell, and then we call it a polar monotrichous bacterium. One great example of this is one of my very favorite types of bacteria. It's called Pseudomonas. It has polar flagella. So they're pretty motile, but not as motile as some bacteria that have many flagella covering the entire surface. So some other ways in which flagella can be distributed, they can be amphitrichous, meaning that they actually come out of both poles. And on Tuesday in lab, when we look at a demoscope of a flagellum stain, we're actually going to see an amphitrichous bacterium where the flagella come out of both ends of the cell. Now our good old friend Helicobacter, it's what we call lophotrichous because it has a tuft of flagella coming from one end of the cell. And then something like E. coli, if it's a motile species of E. coli, will have peritrichous flagella, meaning that it entirely covers the surface of the cell. I actually think I have an E. coli up in my stuffed microbes. They didn't do the perfect job with this because they're showing a lot of flagella on the surface, but they tried to make it look like hair, and really that wouldn't be the case, right? It'd be covering the entire surface of the cell. So poor stuffed microbe E. coli needs a little bit of cosmetic reconstruction, more body hair, so to speak, covering the entire surface of the cell. So this is distribution of flagella. We might then ask the question of what causes these flagella to rotate? That is, what in the world inspires bacteria to want to move from one place to another? And I know if we were in class right now, I would see hands going up when I asked what inspires that in the environment. Because food, right? Food would be one clear answer. Bacteria want to move towards nutrients. But what do they want to move away from? They want to move away from waste, right? Things that are repellents. So they want to move towards attractants, food, away from waste or repellents. And that is partly what's going to dictate their motion. So their motion will be one of two types. You're either going to see the flagella moving clockwise or counterclockwise. The counterclockwise motion is going to cause a run. It's going to move in a direction towards something. The clockwise rotation is going to cause a tumble. So these are the two ways a bacterium can move. A run, a tumble, a run, a tumble, a run, a tumble. And that's how, in a very indirect way, it gets somewhere. So again, comparing to Grant when he's out running, he's going to go from point to point. He's going to say, you know, I really feel like running from Laramie to Happy Jack today. I'm going to just run up Pilot Hill. Great. He's going to do that spatially. Bacteria don't sense that way. It's not like they're going to say, I'm going to go straight from this spot to this spot. Instead, they sense temporally. That is to say, they say, you know, here I am at one spot, and I sense it, and then I have to move to then sense the next spot. So they have to change positions to sense a change in condition. That's called temporal sensing. So chemotaxis, the process of moving, right, taxis is moving, in response to chemicals, towards an attractant, away from a repellent. So toxins, things like waste products, those are all going to be repellents. Food, maybe for some bacteria, light may be an attractant, right? It depends on whether they're chemotactic or phototactic, but they're moving in response to something that they want to get to. So they do this by comparing the current environment with the environment they were in a few seconds before. That is, it's a time-based comparison. So another example, you know, we know Eric is a storm chaser, so let's say that he's sitting down watching this lecture while at the same time keeping an eye on the weather outside. And he feels this draft of cold air, looks up, and he's like, it's getting cold outside, there's going to be a crazy storm, I hope, fingers crossed, right? And he senses that spatially because he can sense the cold draft over here. But a bacterium could not do that. A bacterium would have to actually keep moving to sense the change. So sometimes I feel like a bacterium might have to keep moving in order to keep thinking, but that's not the case, right? We sense spatially and they sense temporally. They have to keep moving from one point to the next so that they can compare time-wise where they were here to where they were a few seconds ago. So the chemical attractant or repellent binds to chemoreceptors. That triggers this whole cascade. Some of you will recognize things like methylation, phosphorylation. Those are events that take place to then allow the proton motive force to swish in and dictate the control of the flagella, the movement of the flagella. So this chemotactic process is what gets bacteria from one place to the next. In the absence of a chemical gradient, they don't move towards anything or away from anything. But remember that they have to keep moving in order to tell if there's ever a change. If they don't, they'll stop sensing. So they have to keep moving, and they will keep doing so in a random way. That is, they'll run, they'll tumble. They'll run, they'll tumble. Run, tumble. And finally, we get this motion that is just completely random. It's not going anywhere. But if after one of their tumbles they suddenly sense a difference in the environment, that is, temporally they say this environment is different than it was before, then they're going to start elongating their runs and lessening their tumbles. This is something called a biased random walk. So when there's a chemical gradient, the length of the run increases, the number of tumbles decreases, and a bacterium is able to get from here to there albeit via a very convoluted path, but it gets from here to there by increasing those runs and eventually finding its way to wherever the chemical attractant is. So at this side of the arrow, that's showing where the chemical attractant is, and the increasing green color shows you that the concentration is getting higher. From this area, the chemoattractant is very low, to this area where the chemoattractant is very high. So that biased random walk is what that is called. Maybe I'll jot that down in case you're interested, that a biased random walk is the way they move. Now you might be thinking, well, what if there's both a chemoattractant and a chemorepellent over here? How does a bacterium choose? What does it choose to respond to? And it's all concentration based. So the concentration wins out, except for that bacteria also sense chemoattractants at a lower concentration than they sense chemorepellents. Sometimes concentrations as low as 10 to the minus 8 will actually be detected by bacterial cells. So they're going to respond to attractants somewhat preferentially because of that. There are also double the number of chemoattractant receptors than there are chemorepellent receptors. So you can see how really they've been somewhat biased to responding to chemoattractants in that way. Awesome. Let's go ahead and stop our coverage then of flagella. And sorry, you should write that down, right? They move towards the attractant. And we're going to turn our attention in the last portions of our coverage for this exam, right? So I'm about ready to cut off the exam coverage. We're going to talk about another structure external to the cell wall called Fimbriae, or sometimes also called Pili, or sometimes these structures are either called Fimbriae or Pili, and I'll specify that in just a minute. So Pili and Fimbriae kind of look like flagella in that they are these hollow cord protrusions external to the cell wall, but they're much shorter and much thinner than flagella. So in the picture that we see here, you can really tell the difference. Right here, and I'll kind of highlight it in blue, this is a flagellum. So seeing that structure there that's very long, that's a flagellum. But then here are the tiny Fimbriae. So you can see these kind of hair-like structures protruding from the cell. Those are the Fimbriae. Now, some textbooks use those two terms interchangeably. They use either Pilis or Fimbriae. Our textbook uses Fimbriae to dictate anything that is not involved in conjugation. That is to say, Fimbriae will refer to anything that is not a sex Pilis. That's how our book uses those two terms. But bear in mind that different texts are a little bit different on that. So these are composed of protein subunits arranged helically, just like within the filament of a flagellum, to form a hollow cord fiber. These allow for a lot of different things. One of those is the adherence to specific surfaces, so allowing it to attach. But then also, often these are involved in a really interesting kind of movement. This movement is called twitching motility, and it's literally the motion of a population of cells across a solid surface. So this sort of twitching motility is common in certain kinds of bacteria. One good example are the myxobacteria. They're important bioremediants and important environmental bacteria. They're moving across surfaces with this short twitching motions. Sometimes because of that, they're given the name wolf packs, like they're moving as a community of cells like a wolf pack. So it's kind of a cool occurrence that you see. The other thing that these can assist in is obviously, attachment, and but lastly, in terms of conjugation. So Pili is the name our book gives to those involved in transfer of genetic material. So the ability of a donor cell, to attach to a recipient cell. And remember how we talked about that allowing for the transmission of particularly plasmid DNA, though sometimes other DNA can be transferred that way as well. So sex Pili, one specific type of these structures that's involved in that genetic transfer of information. This ends your coverage for the first exam. This is the last thing that will be fair game to be tested on for this first exam. Everything that we cover from here on out will not be included on the exam.